Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for a speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions from the chat window. Your questions in the chat window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Geert van Raemdonck from PharmaFluidics and Koen Sandra from RIC.
Thank you, Leah, for the kind introduction, and thanks again for giving us the opportunity to set up this webinar to present and explain our innovative approach to make liquid chromatography columns. I also want to thank the participants for their time and interest into our products and technology. This is what we will covering today. First of all, I will give a short overview of the Micropillar array column technology. Then Dr. Koen Sandra from the Research Institute for Chromatography in Belgium will take over and cover the benefits that our µPAC columns can bring to host cell protein characterization. Finally, we will both be available for answering any question you may have. At PharmaFluidics, we use a fundamentally different approach to make chromatography columns that allows introducing perfect order into the separation bed. These columns are called Micropillar array columns, or shortly µPAC columns. Let's first have a look at the design of traditional packed bed columns. The stationary phase of these conventional columns consists of a bed of microparticles that have been packed into a cylinder or a capillary tube. These particles have a certain size distribution and thus consequently have a certain degree of heterogeneity. On top of that, their location in the column is more or less random, at best not perfectly ordered. As illustrated in the bottom figure, this disorder will lead to additional dispersion as the sample migrates through the column. The fact that not all flow paths in the column are identical will cause sample molecules to have slightly different retention times, causing additional dispersion. Besides, we think that packed bed column technology gradually reaching its limits, more specific in terms of what can be achieved by reducing particle diameter and by further increasing the column length. PharmaFluidics uses a fundamentally different approach to fabricate their chromatographic columns. Instead of using particles that are packed into a tube or capillary, the backbone of the stationary phase is designed in a similar way that electronic circuits or sensors are designed. State-of-the-art lithographic techniques, followed by silicon etching, are used to realize microfluidic channels into a silicon wafer. These channels are filled with accurately positioned ordered arrays of pillar, which form the backbone of the stationary phase. This results in two important benefits. Virtually perfect order is achieved, whereby reducing the eddy dispersion to an absolute minimum. This is again illustrated in the bottom figure, where you can see that all sample molecules will follow identical flow paths throughout the entire column. The second benefit of its format comes from the fact that the pillars are freestanding and positioned at an accurately defined distance from each other. This has a serious impact on the column permeability and enables us to provide very long columns that can be operated at moderate pressures. The benefit of order versus disorder is clearly illustrated in the animated computer simulation on the right that have been generated in Professor Geert's group, and that are formed the basis for the current µPAC columns. In the disordered case, the sample plug is gradually dispersed as it migrates through the stationary phase, resulting in significant peak broadening of the sample plug. This will affect the separation, resolution, and cause dilution of the sample, whereas in the ordered case, the sample plug remains very nicely conserved and the intensity remains high, resulting in improved resolution and high signal-to-noise ratios. To make long columns on a limited footprint, the columns need to be folded. The current production process is based on a 4-inch silicon wafer, one of which is shown in the middle picture, and on such a wafer we realize 3 2-meter columns and 2 50-centimeter columns. A 2-meter column actually consists of 40 discrete lanes that are interconnected by 180-degree turns, finally resulting in a 200-centimeter long meandering or serpentine channel. Making microfluidic turns without introducing additional dispersion is quite difficult, because racetrack effects, where the inner lane is shorter than the outer one, need to be avoided. To avoid these effects, we use proprietary flow distribution and collection structures at the beginning and the end of each lane. These are shown on top. At the bottom of the slide, we gradually zoom in up on the pillar surface from right to left. Once the pillar structures have been etched, they are rendered superficially porous to increase the effective interaction surface. The final result are channels filled with 5 micrometer diameter pillars positioned at a distance of 2.5 micrometer with a height of each pillar at 18 micrometer. The porous layer round is 300 nanometer thick and the average pore size is estimated to about 10 to 20 nanometer. The bonded phase for the current products is reverse phase C18. A feature that was very helpful during the research and development phase is that our columns have a transparent glass cover, allowing us to follow what happens inside the column using fluorescent dyes. The first screenshot shows a real-time injection of such a fluorescent dye into the column. It illustrates nicely how the injection is controlled by the flow distributor in order to uniformly distribute the sample over the relative wide separation bed, and how the separated bands remain nicely straight and parallel. The second video shows how the proprietary collection and distribution structures allow to conserve the sample plug in 180-degree turn. Which we see here is not a real-time separation, but instead a number of sequential low-volume injections. By combining these two principal capabilities, we can provide long columns that maintain sharp peaks. Nowadays, our µPAC columns are frequently used in comprehensive proteomic research, such as biomarker discovery, where rare molecules are sought in complex backgrounds. However, on the other hand, is the separation performance of our columns also key to detect subtle differences and modifications for biopharmaceutical applications, such as biosimilar development, and for host cell protein analysis, like further discussed in this webinar. At the moment, we offer 2 different lengths for our analytical µPAC columns. With a column volume of 3 microliter and a flow rate ranging from 0.1 to 2 microliter per minute, the 50 centimeter µPAC column is ideally suited to perform more routine analysis where increased throughput is needed. To serve those who are aiming at comprehensive proteome coverage, PharmaFluidics offers a 200 centimeter long µPAC column with a volume of 9 microliter and a respective flow rate ranging from 0.1 to 1 microliter per minute. In addition, we are very delighted to announce you our latest product, which is a 50 centimeter µPAC C18 CAP-LC column. Since this column was developed according to the proven µPAC technology, it offers some unique features, like unrivaled separation power, exceptional high reproducibility through the micro-machined fabrication procedure, easy to connect and to integrate through the 1/16 of an inch connectivity on both sides, and a high flow rate flexibility ranging between 1 and 50 microliter per minute. This makes this column ideally suited for scientists who are looking for high throughput and increased robustness without losing sensitivity. Our µPAC CAP-LC column will be launched at the end of this year. This closes my part of the webinar covering the µPAC column technology. I am very pleased now to hand over the floor to Koen Sandra from the Research Institute for Chromatography, who will share his experience about working with our µPAC columns to analyze host cell proteins.
Okay, thank you, Geert, for this very nice overview of the µPAC technology. So in the next 10 to 12 minutes, I will demonstrate, in fact, how we at RIC have used this technology in host cell protein monitoring, and more specifically during the downstream processing of a therapeutic monoclonal antibody. Now, before showing the performance of the column in host cell protein analysis, I would like to start with providing you some more context here. Now, as most of the people in the audience know, monoclonal antibodies are tetrameric macromolecules with a therapeutic effect that are typically being used in the treatment of the more severe diseases, such as cancer and autoimmune diseases. Now, today, these products are really dominating the pharmaceutical landscape. And in fact, over 80 of these products have been approved for therapeutic use already in the US and in Europe. And over 550 of these products are currently in clinical development. And important to mention here as well is that around 62 are in late stage development. Now, in 2017, these products had a total sales value of around $123 billion, and this represents around 65% of the total biopharmaceutical market. So this is really a substantial amount there. And monoclonal antibodies are also considered, in fact, as the fastest growing class of any therapeutics. Now, monoclonal antibodies are commonly produced recombinantly in mammalian expression systems such as CHO cells. And of course, next to the therapeutic antibody, these cells, they also produce endogenous host cell proteins, abbreviated as HCPs. And these HCPs, in fact, can contaminate the therapeutic, this despite multiple purification steps that we have throughout the process. Now, since these process-related impurities can affect product safety and efficacy, of course, we need to closely monitor these products. Now, today, Eliza is in fact recognized as the gold standard technology for measuring these HCPs, and this is a result of the high sensitivity and also the high throughput of this methodology. Now, Eliza, however, does not recognize all the HCP species, and it only provides information, in fact, on the total amount of HCPs, and it does not provide any insight in individual HCPs. Mass spectrometry, then again, is in fact gaining acceptance as an alternative and complementary technology for host cell protein analysis because it can provide both qualitative and quantitative information on individual host cell proteins. Now, MS-based host cell protein analysis typically relies on bottom-up proteomics. So this means that we in fact handle peptides that are derived from the protein, so typically following cryptic digestion. And as a result, one is of course confronted with an enormous complexity and also an enormous dynamic range. And the separation space is really dominated by peptides that are derived from the therapeutic protein. Now, if we then, of course, want to successfully analyze these host cell proteins, we really need highly efficient separations upfront mass spectrometry. And in that respect, the Micropillar array columns are extremely promising. And as just discussed by Geert, due to the order that exists in these columns, the peak dispersion can really be minimized, and highly efficient separations are the result of that. And this really allows for in-depth host cell protein monitoring. Now, we at RIC have used the µPAC technology in combination with a Q-exactive HF mass spectrometer for the characterization of host cell proteins throughout the downstream processing of a therapeutic monoclonal antibody. And the therapeutic monoclonal antibody I would like to discuss has been recombinantly expressed in CHO cells. Now, what we did, samples collected at different steps in the process were reduced with DTP, alkylated with uriacetamide, and afterwards digested using trypsin. and then the resulting peptides were separated on the MicroPAC column. Now for that, in fact, we loaded 2 micrograms of the digested sample on a 2-meter long MicroPAC column, and in fact loading took place via a MicroPAC trapping column. After the loading, the peptides were then separated using a 160-minute gradient, making use of a mobile phase combination of water, formic acid, and acetonitrile. Now the flow rate on the MicroPAC analytical column was 500 nanoliter per minute, and during loading, in fact, the flow rate was 10 microliter per minute over the trapping column. Now, during the separation, the Q-exactive mass spectrometer was continuously acquiring MS and MSMS data in the data-dependent acquisition mode. And in fact, one survey MS scan was combined with 10 dependent MSMS scans. And then all the data, all the MS and the MSMS data were subsequently searched against the show database that was extended with the sequence of the therapeutic product. And the table shown in this slide shows, in fact, some important metrics that we obtained on the different downstream process samples. So in the harvest sample, we could acquire close to 70,000 MSMS spectra, and after database searching, close to 30,000 unique peptides could be identified, and this corresponds, in fact, with 1,812 proteins with more than one PSM or with more than one peptide spectral match. So these are 1,812 confidentially identified proteins. And what we see, in fact, is that throughout the purification process, that we have a very nice clearance of these host cell proteins, and in the last purification step, the number of host cell proteins could really be reduced to only 2. Now, semi-quantitation of all these host cell proteins was done by spiking 4 pre-digested proteins at known concentrations in every sample. And afterwards, the MS abundance of these pre-digested proteins was linked to the MS abundance of the identified host cell proteins. And in this manner, we could in fact determine a concentration for the host cell proteins expressed in ppm. Now this slide here plots the number of host cell proteins that we detected at a given concentration in the different downstream processing samples. And in the harvest samples, we see in fact that host cell proteins are detected between 1 ppm and 600,000 ppm. And what we also see that throughout the process, we do observe a quick drop in HCP concentration to below 100 ppm to then end up in the final sample below a value of 10 ppm. Now another interesting view into the data is presented in this slide here, which shows in fact that in the harvest sample, the total host cell protein concentration is at 2 milligram per milligram of protein. So this is 2 million ppm that we are talking about here. And in the final purification step, the total host cell protein concentration drops drastically to only 9.9 ppm or 9.9 nanogram per milligram of therapeutic protein. And in both cases, the therapeutic is quantified at 1.3 milligram per milligram, where it theoretically should be 1 milligram, of course. And this gives you already a very nice idea on the accuracy of this semi-quantitation strategy, which is generally expected to be between 50 and 200%. Now, the bar plots that are presented in the current slide follow, in fact, 2 specific host cell proteins throughout the process. Now, on the left, we have clusterin, and clustering, for example, is present at 59,000 ppm in the harvest sample, and it could be reduced down to 3.1 ppm in the final purification step. Now, clusterin has already been described to easily associate in a non-specific manner with both the Fc as well as the Fab fragment of antibodies. So it's not a surprise to see that host cell protein further down into the process. Now, another protein that we detect is the phospholipid transfer protein isoform X2, which is present at 2,500 ppm in the harvest, and it in fact reaches 0 ppm in the final purification step. Now, to put things somewhat more in perspective, This slide here shows the extracted I chromatogram of one of the peptides corresponding to the host cell protein that we detect at 3.6 ppm. And here it is shown, in fact, in overlay with the base peak chromatogram. And this nicely shows the dynamic range that we are confronted with. So despite the fact that this particular host cell protein is only at 3.6 ppm, we can also obtain some very nice MSMS data as is shown here, and this of course allows us to confidentially identify this protein. Now when we also consider the base peak chromatogram, it in fact becomes clear that the column is overloaded. So we do have some very broad peaks, and these broad peaks that we observe, they can all be traced back to the therapeutic product. Peptides originating from the host cell proteins, on the other hand, they give nice Gaussian and sharp peaks, as in fact shown here in the inset of this chromatogram. And of course, if we want to detect host cell proteins at high sensitivity, this overloading is really required, especially if we want to go down to 1 ppm. But in fact, the µPAC column can clearly handle this overload. And in some specific cases, we could even cross the 1 ppm barrier, which is in fact the ultimate in host cell protein analysis. And with this statement, I would like to come to the end of my part of the presentation. So I hope I have been able to convince you that micropillar array columns, they do really represent a valuable novel tool for the characterization of host cell proteins and their monitoring throughout downstream processing. And in the harvest sample, we could demonstrate that we can detect close to 1,900 proteins. They could be all confidentially identified, and we could also quantify proteins readily down to 1 ppm. Now, if this would not be satisfactory for your purposes, so if you really want to go below that 1 ppm value consistently, well then content and also sensitivity can further be improved by implementing a second chromatographic dimension. So this brings us into the area of two-dimensional liquid chromatography, but we can also optimize sample preparation, for example, by performing a depletion of the therapeutic antibody, for example, implementing a protein A affinity step, or we can also do host cell protein directed digestion. Then again, we can also do some optimization at the mass spectrometric side, for example, by working with exclusion lists of peptides that are originating from the therapeutic, but we can also operate the mass spectrometer, of course, in the data-independent mode. So all these things could eventually improve the ACP content and sensitivity and bring it below 1 ppm if that would be required. And with this, Geert, I would again like to pass the word over to you.
Perfect. Thank you very much, Koen. Now we're almost at the end of this webinar. However, first I would like to thank our collaborators from RIC to perform these experiments and to process the data. Further, I want to thank also my colleagues from PharmaFluidics for all their hard work and efforts, and of course Professor Geert de Smet, as he is the intellectual father of this development, and Professor Raemdonck, who translated the theoretical models into practice. Finally, we would like to thank also the people from Thermo Scientific who gave us the opportunity to use their high-end instrumentation on several occasions. If you want more information about our products, you can find all application notes and contact details on our website or send us an email at info@pharmafluidics.com. As already mentioned, we have currently two types of nanoflow columns on the shelf, a 50 cm and a 200 cm long column, while our CAP LC column will be launched at the end of this year. Thank you very much for your attention, and we are happy to try answering all your questions.
Great, thanks. So we do have some questions. The first question is, do the pillar array columns clog, especially with overloading?
Due to the high permeability of 2.5 micrometer, our µPAC columns are less prone for clogging, so they have a higher permeability compared to the conventional packed bed columns. Of course, it's always possible to clog any system, but the risk is less with the µPAC technology.
Another question, the results you presented were on a TMO Q Exactive. Are there similar results available on other MS instruments?
Yes, we performed already some experiments on an timsTOF Pro instrument from Bruker, where we could also find some very nice results. So more information will be available on our website later on.
Okay. And did you validate the HCP findings with Eliza?
Yeah, so we have done ELISA measurements using a commercial host cell protein kit. We performed ELISA measurements not on the harvest, but on the samples that were further down in the process. And in fact, our ELISA measurements nicely correlate with the MS data. But this is not always the case. So in our case, we have a nice correlation, but this is of course not always the case. And for this reason, it is highly valuable if you can combine both host cell protein ELISA with mass spectrometry, because very often you will get in fact complementary information, and you can also confirm the performance of the host cell protein ELISA with mass spectrometry.
Okay. And how do you quantify DDA data?
Yeah, so that's an interesting question. So for that, we use the MS trace. So we make sure that we have sufficient MS data points, because we need to quantify on the MS level. And for that, we of course we quantify against 4 pre-digested proteins using the high-3 label-free quantification approach. So we do calculate a universal response factor, and then we quantify every host cell protein against this universal response factor. But take into account, we do quantify based on the MS data.
Okay, and I think this is a follow-on question to that. How do the columns perform in terms of reproducibility?
Yeah, so the fabrication process of the columns, they're made by chemical etching. So the same mask is used to produce every column. So every column or the pillars are placed at identical positions. So every column is identical the same.
And do you use ion exclusion lists for MS?
In this case, we did not, but this is of course an interesting option to increase the host cell protein content. So you could use an exclusion list for your therapeutic peptides, and in fact you can also perform subsequent runs where you also exclude the host cell proteins peptides that were identified in an earlier run. So this is definitely a very interesting option to follow up further.
And have you done any HCP work done on the NIST MAB? And if so, how many HCPs were identified?
Yes, we performed already some experiments on an instrument, on the timsTOF Pro instrument on the NIST map. It was a slightly different approach where we used a sample preparation according to the Lilly paper from Huang et al. And there we could identify 316 protein identifications with 1 peptide and 91 protein identifications on at least 3 identified peptides.
Okay, so I think we have time for just one more question. If we didn't get to your question, those will be passed on to our presenters and they'll be able to follow up with you individually. So the last question is, how many injections can I use in one Micropillar pack column?
Yes, that's a little bit that's a difficult question. It's hard to predict, but we performed an experiment in-house and we ran the column for 6 months on an instrument, and we performed more than 3,500 injections without any loss of performance. And nowadays we have also some core labs that work with our columns that can confirm these values. More information about this experiment can be found in an application note on our website.
Okay, well, thank you, Geert and Koen.
Perfect.
Thank
Thank
you.
you very much.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.